Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there.
Amino acids are used to make proteins. Table 1 shows the R groups of 6 different amino acids. Well, there you go. So, use table 1 to identify 3 different amino acids used to make the polypeptide shown in this figure. So, what we need to do is we've got to look at like how amino acids just vaguely sort of work. So every amino acid in the universe has like a C, an H, an R group, which can change, a carboxyl group COOH, and a naming group H2N. So whenever you're seeing a C next to an N and a C like that, that's probably the core amino acid you've got. And then obviously these can vary. So this H can disappear and that can gain an H or so. Okay, so we need to understand what the general structure of an amino acid is. So you've got a C, an R group, an H, and then a COOH, the carboxyl group, and a naming group, so H2N here. So these can vary and change about and stuff to link with other things. And if you see that there's a carbon next to a nitrogen and another carbon here, and then there's an R group up here and an H, something that fits this sort of look, then you know what that's an amino acid. So here it says it's a polypeptide in this whole thing. So we need to look for where that sort of CCOH thing is and what, it could be. So here's a C, here's a C, H2N. If you look, that's probably linked there. So you've got a C double bond O. So that's like your sort of one amino acid's core. There's an H there, that has to be the R group. So this left amino acid, its R group is that, which they've told you in the question is serine. Like so. Now if you look at the middle one, so again, we're looking for where the CN is and the C double bond O next to it, like part of the COH group, which would be here. And then there's an H here and there's an R group here. So this middle one's R group has to be CH3, which is alanine. And then if we look at the very last one, so again, we're looking for the same pattern. So we're looking for a C bonded to an N and a COH and an H. So that's the core of it, I guess. And then if you want to call it that. And then there's the R group. So that last one is aspartic acid. So table 3 shows 3 statements and the names of 4 biological molecules. Put a tick next to the box when the statement is true for each for the biological molecule. So peptide bonds, they are formed between amino acids. So whatever you've got that's got amino acids bonded to each other will have peptide bonds. So DNA is no, ATP is a no, reverse transcriptase is an enzyme which is a protein and that's made of amino acids, so yes, and phospholipids no. Then it says it's formed using a condensation reaction. So every single bond that you've covered, so glycosidic, ester, phosphodiester, and peptide, are all through condensation reactions which a molecule of water is released. So DNA is a yes, a phosphodiester. ATP as well is a yes because there's a group between those phosphate groups, the bond between them too. Reverse transcriptase as well, yes, and phospholipids, yes. Is a polymer. So a polymer, remember, by definition, is where you've got repeating units of the same thing over and over and over and over and over again. So lots and lots of amino acids. So this is a polymer of nucleotides, DNA nucleotides. So that's a yes. ATP is not a polymer because you've only got, adenosine and 3 phosphates, so no. Reverse transcriptase, remember that's an enzyme, which is a protein, which is a huge amount of like amino acids, bonded together. So yes. And then phospholipids aren't as well because you've got a glycerol, 2 fatty acids, and a phosphate group, so it doesn't count as a polymer because it's not repeating unit. So figure 2 represents the structure of adult human hemoglobin. So we've got beta chains and alpha chains, and the beta chains are gray, the alpha chains are white. So the number of amino acids in each of the beta chains is 3.546% greater than the alpha chains. Each alpha chain contains 141 amino acids. So, right, AAs for short, is going to be 3. So that's like saying if you've got 3.546% more than something, then you could say that's the same as like 103.456%. So 103.456% of the alpha, because you've got the whole of alpha but an extra 3.456% on top. So they've told you that each alpha chain has 141 amino acids. So that means that beta is going to be 103.546 over 100, because we're doing percentages. If you were to find like 90% of something, you did 90 over 100 times 141. So that must mean it gives you 145.99986. But because there's 2 of them in this hemoglobin molecule, we need to times it by 2. So if we do that, we get 291, right? But we're not done because they're asking you to find how many amino acids are in total of the entire hemoglobin molecule. So we've got 291 for the betas, and then they've given you the alpha as 141. So you need to also do that times 2. So 141 times 2 gives you 282, and then you add them together. and that gives you 573. So when a substance called BPG binds to hemoglobin, it reduces the affinity of hemoglobin for oxygen. So if we look at figure 3, it shows you an oxyhemoglobin dissociation curve for hemoglobin in normal conditions, and it's asking you to sketch what happens to the curve when you've got BPG bound to it. So I ain't gonna lie, I don't sit and bother remembering, oh, higher affinity means to the whatever side, left or right or whatever. I just have a think about what's going on. Normally, you in your lungs, these are saturating like crazy because there's loads of oxygen. That makes sense because percentage saturation of hemoglobin means how much hemoglobin is bound to oxygen. As you start to decrease the partial pressure, you're going from the lungs where it's high to respiring tissues. So expectedly, it dissociates like that and it goes down. So if you've got what did it say? It's a lower affinity. Lower affinity, in simple terms, means that hemoglobin, the hemoglobin doesn't hold on to oxygen as strongly. It lets go of it more readily. So what happens is, you just imagine the curve. It's like, oh, it's holding onto it, it's holding onto it, and then it just drops sooner. It lets go of the oxygen sooner, and there you go, like that. That's it. It should level off at the same points there, and that's all it is. It's a lower affinity is how it does it, and that makes sense because again, it's letting go of it more quickly. Whereas, for example, if it's high affinity, it's holding onto it a bit more, and then it drops like that. So it says, suggest and explain when it would be an advantage for a human for BPG to bind to hemoglobin. So again, like it said before, it's told you that if you do that, you have a lower affinity of O2. People word, so that means is it lets go of the O2 easier. So you need to think, why is it letting oxygen go earlier a benefit? Well, think about what you need for oxygen. You need it for respiration, and think about something where hemoglobin and blood delivery to things requires oxygen and stuff. So during exercise, there is more O2 released to aerobic, I'm gonna write aerob for short, respiring tissues. And there you go. Okay, so figure 4 shows the diagram of a Paxinian corpuscle. Name the structures P, Q, and R shown in this diagram. So R is it's the Schwann cells, or you could say myelin sheath. Either is fine. So R, where are we? Schwann cells or myelin sheath. Whoops, it's gone off the thing. Q then, what is that? Well, this is like your sensory nerve ending, which they do accept actually. Or it's the axon, if we want to use the technical terms. It's the axon's membrane. Next, P. Well, P is the lamellae that surround it. Or you can say capsule as well. Either's fine. In order to do this question, it's helpful to think about how the Paxinian corpuscle works. It helps us determine our sense of touch. This is a free sensory nerve ending with your stretch-gated sodium ion channels. These lamellae make sure that it can stretch and mess around with this. And then obviously it's a nerve, so it's going to have its usual sort of myelin to get to the nervous system to make it saltatory conduct quicker. So if you think of it like that, it helps make sense of why it's built the way it is. Because these lamellae deform and stretch and stuff, which then deforms and stretches this. So now it says, 2 students A and B investigated reaction time in response to touch. So student A sat with her eyes shut and her forearm resting on a worktop so that her hand was over the edge. Student B held a ruler vertically between student A's thumb and first finger with the ruler at 0 millimeters, lightly touching the student A's first finger. Student B released the ruler, and as soon as student A felt the ruler fall, she closed her thumb and the first finger to catch the ruler as shown in figure 5. Student B measured the distance the ruler had traveled to, that had fallen to the nearest millimeter. So what we got, they're just about holding it. Student B lets go and they catch it. So the test was then repeated 3 more times using the same hand to catch the ruler. Table 3 shows A's results. So just have a quick look to describe to yourself what the results are showing. So trial 1, 2, 3, 4, the distance the ruler had fallen was quite variable actually. From 79 to 97 to 10 to 94. One of them looks a bit out of place, but let's see what happens. Then it says the student was able to convert these distances into reaction times using table 4. So we got the distance that the ruler had fell, 10, 20, 30, and so on. And then the reaction time as well. So again, describe the pattern. So as this distance is falling, increases, it looks like the reaction time is also increasing as well. Cool. So now it says, calculate the percentage uncertainty of the measurement of trial 1 in table 3. So if you look at trial 1, they've measured 79 as their measurement. It's a millimeter ruler, so the uncertainty is going to be plus or minus the smallest increment, which is 1 millimeter. So if we do that, we get, to work out the percentage uncertainty, it's going to be your smallest increment divided by the actual measurement you took, which is going to be, and then times by 100, which gives you 1.27. or 1.265 and so forth, and that's going to be this one here. In this investigation, it's not possible for a student to react in less than 45 milliseconds. Suggest one explanation for the value recorded in trial 3 and table 3. So what they're saying is like, you cannot go below, what was it, 45 at all. So how on earth did they get 10? It's not like they got some superhuman thing. What it's implying, the only reason it could happen is probably because something went wrong with the experiment. So we need to think about what's going on here then. So again, person's hand is chilling, they drop the ruler, and then they grab onto the ruler. So you need to think about anything that would stop it from falling the same distance, which means it must have, what's the word? They must have reacted too early. So you've got to think of a way to put that into words. So what could have happened is that the student may have closed her hand before the ruler was released. Additionally, you could say things like, well, I mean, first of all, they're holding it, so it could have stuck to their skin. It could be that it didn't fall vertically. It might have smacked the side of their head or something. Things like that as well. But I think that's pretty, like, a good one. So student A estimated that the length of the nerve pathway involved is 175 centimeters. So nerve pathway is 175 centimeters. It says use table 3 and 4 to calculate the mean speed of nerve impulse transmission and do not use the value for trial 3. So if we look at the average distance fallen, that's going to be 79 plus 97 plus 94 divided by 3. So 79 plus 97 plus 94 all divided by 3 gives you 90 millimeters. And then if we look at we got this 90 millimeter as our distance that we've fallen. So then what we do is we look at this table here to find out the reaction time we need. 90 millimeters, 136 seconds. So the reaction time is 136 milliseconds, and they want it in meters per second. So if we convert that to seconds, it's milli to seconds divided by 1,000. It's going to be 0.136 seconds. And the nerve pathway is 175 centimeters, which is going to be 1.75 meters. So then it says meters per second, so it's a meters divided by seconds thing, so distance over time. So d over t, which is going to be 1.75 divided by 0.136, which gives you 12.86, or roughly 12.9. And there you go. That's how you do that one. In response to touch, nerve impulses can be traveled at speeds of 76.2 meters per second. Suggest 3 reasons why in this investigation the estimated speed of student A's transmission was less than 76.2. So again, the speed is going to be dependent on the distance over the time. So speed can get screwed over by the distance or the time. Now in this case, they've estimated that length. So there's not really anything you could think about with regards to doing estimates of the nerve pathway. It's more to do about what they did in this experiment, because this experiment doesn't say anything about estimating the nerve pathway and how they did it. It just says the student had an estimated value. So it's telling you to critique this experiment, and the only thing that can go wrong in this experiment are things to do with the timing. So, or like factors that need considering with the timing. So what might have happened is that, for example, if you think about it, you're dropping it, you're going to have you've got to grab the ruler. It's going to take a second for the actual muscles to contract. So that could be one of them. So time for muscle contraction. So it could be that they're dropping it and it's not like a reflex. It could be like they have to actually think about it, be like, oh yeah, I have to grab this. So this time taken for, the brain to coordinate muscle contraction. Because again, it's not like a reflex. They have to think about it actively. What else can we think of? So as well as that, it could be, for example, that they need to feel the actual ruler passing across their skin, and then it goes via sensory nerve to the brain and so forth until they have that muscle contraction. So it might be that there's also time taken for the Paxinian corpuscle to open, stretch mediated sodium ion. You can't say sodium, you have to say sodium ions or Na plus ions channels. So to open those, because then you can say, well, the speed is, again, it's dependent on time. So if you increase the time, because it's on, you're dividing here, it's on the bottom, that's going to make the speed go down. So because they've told you that the speed is lower, it must mean that the time's gone higher. Because in this other question, you can't talk about the estimates of the nerve pathway because there's no, it's not telling, that's not mentioned in the experiment how they did that. So a student prepared a stained squash of cells from the root tips of garlic to calculate mitotic index. What they did is they cut the end 5 centimeters from 10 garlic roots. They placed the root tips in a petri dish containing 5 centimeters cubed of hydrochloric acid for 12 minutes. They rinsed the root tips in distilled water. They placed one of the root tips on a microscope slide and added toluidine, whatever, blue stain. They placed a cover slip onto the microscope slide and gently pressed the cover slip downwards onto the root tip, and then they observed them using an optical microscope. Suggest why the students soaked the root tips in hydrochloric acid in step 2. So, we're dealing with plants, because they've told you it's with garlic. If we put this stain in, it's not going to be able to get through into the cell and stain what it needs to do. So, we need to figure out a way to actually allow it to get in. So, one of these reasons is to actually break the cell walls. So, it's to break, or hydrolyze, I guess I should say, better word, the cell walls. So, because remember, you got to say your direct consequence, the stain can diffuse into the cell. As well as that, if we're looking at mitotic index, we don't want them to carry on doing mitosis while you're counting it. Otherwise, while you're counting one, the other one's still doing mitosis, and you'll get you'll get a higher rate of mitosis. So you want to also stop mitosis. You want them to just be done so that you can look and it's almost like freezing them in that sense, and you can look at what they're, just about to start doing. Pressing the cover slip downwards enables the student to observe the stages of mitosis more clearly. Why? So the reason is because when you're pushing it, you're spreading the cells out, you're squishing them out to the side, and it gives you a thin layer. So it's to create a single thin layer of cells, and then you might, you got to think, well, why? They're using an optical microscope. Why would a thin layer of cells be a good thing? Well, it's to let light pass through. Because if you had like a really, really like thick layer of cells, the light can't get through. You won't see them clearly. So it now says Figure 6 shows the students' drawings of one field of view. Name the stage of mitosis shown in cell G and explain the appearance of the cell. So in cell G, you can see that we've got some very distinct-looking V-shaped chromatids. And they're on opposite sides of the cells. So that one's anaphase. And why? Well, like I just said, chromatids pulled to opposite poles of the cell or end. So now it says use figure 6 to calculate a mitotic index for the cells in this view. So the mitotic index is going to equal the number of cells with visible chromosomes over the total number of cells seen. So if we do that, let's just work out the total numbers. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. So we have 39 as our total, and we need to look at how many have got visible chromosomes. So 1, 2, 3, 4, 5. So that gives you 0.128, which is roughly 0. Like 1 or 1.3, you could say. So now it says, other students in the class follow the same methods but got different mitotic value indices. So we need to think about what's going on. Well, if you think of the mitotic index, it's the number of cells with visible chromosomes over the total number of cells observed. Now, if we look at just that formula alone, it could imply that maybe, they just happen to see more with visible chromosomes, or they might not have seen like the same number of cells. But you've got to remember it's an index for that reason. It compares the two. So what you need to do is you need to think about what they looked at in the experiment and how that might have screwed things up instead. So if you look at this guy's method, what he's done is he cut the end of 10 garlic roots and pieces and then, did all of that. So what if, they got plants that, they got roots from different ages or like, different times, like different amount of mitosis. So roots of different ages. Or also you could think maybe, well, perhaps they might have, cut different parts of the root tip. Some parts, parts of root tip. Some other parts might be like, they might do mitosis more than others. And, you could also say that, like, they could do the root cells can undergo mitosis at different times, rates and stuff. It could be that, like, in this question, all we see is that they're doing one field of view as well. So it might be that, one person's field of view that they're counting the cells in might not actually capture all of them, whereas somebody else's will as well. So you've got to say something like, they might have just looked at, other students might have looked at more than one field of view, and this guy's only done one. So now it says, complete the following definitions. The genome is, well, it's the complete set of genes in a cell or organism. And the proteome is the range of proteins a cell can produce. So it's very important you understand these definitions. It's referring to just the cells that this one particular cell can make. So for example, a stomach acid cell makes stomach acid, whereas like a bone cell don't. So the proteomes would vary. Recombinant DNA technology can involve the transfer of fragments of human DNA into bacteria. The bacteria then used to produce human proteins. So give 2 reasons why bacteria are able to use human DNA to produce human proteins. So you need to realize why on earth breeding ATCG from a human can also result in the same thing in bacteria. So number one, I guess you could say straight off the bat, is that the DNA is universal. So one base triplet in one organism is going to be the same base triplet in another. But that's just talking about making amino acids, because it's talking about how you can use human DNA to make human proteins. So again, to make a protein, what are the 2 things that must happen? Pause and have a think. All right, cool. Yeah, so it's transcription and translation. So in order to go from DNA to a protein, you've got to do these 2 things. So that must mean if we're able to put human DNA in and it's universal and we end up with the same protein, then it must mean that the mechanisms of transcription and translation are also universal. Otherwise, why would that give us the same protein? Remember that proteins aren't just like you make this amino acid. Like you got your amino acids, they're bonded together via peptide bonds, and then they eventually fold and coil and so forth. So if you're making the same proteins, not only is the DNA universal, the actual mechanisms that produce the protein are as well. Suggest and explain one reason why bacteria might not be able to produce every human protein. So for example, like I just said before, in order to do that, you need to do transcription and you need to do translation. So we need to compare what that's like in humans versus bacteria. So translation is effectively going to be the same. It's just mRNA gets to a ribosome and all the other stuff happens. So it's probably something to go with transcription that's different. Well, I mean, okay, we can think a bit beyond that as well. But if we look at transcription, humans are eukaryotes, bacteria are prokaryotes. What's the difference you can think of? So in humans, they have introns, so there's no intron splicing in prokaryotes. So that's one of them. As well as that, think about what like proteins go through when they're being made as well, beyond just, oh yeah, the amino acid sequence is made. They've got to be packaged and processed and, shipped out the cell. So as well as that, well, bacteria don't have Golgi apparatus. So for example, glycoproteins, we need those. Those are proteins with like glucosey bits and sugary bits on them. And carbohydrates, well, bacteria don't have this, so they can't do that. So they don't have Golgi apparatus to modify proteins. Because Golgi apparatus is a membrane-bound organelle and prokaryotes don't have those. So antithrombin is a protein and it prevents blood from clotting too much. Some people have a deficiency of antithrombin in their blood, so they need to inject this protein. Genetically modified goats used to produce this protein. The human antithrombin gene is transferred into goat embryos and the adult goats then make human antithrombin protein. Figure 7 shows an example of a DNA fragment that can be transferred into the cells of goats. Whoops. The enhancer stimulates the region M. Name the region M and N shown in this figure. So we've got an enhancer, some mysterious M, a marker gene. So the marker gene will tell them, hey, this is the gene we want to be mucking around with. So, it could make it fluorescent, for example, so they know that's what it is. Then you've got the actual antithrombin gene. So remember that genes code for protein. So this is the actual protein bit that's going to lead to your antithrombin protein. And then we've got region N as well. So what could be going on around, coincidentally, right next to M and N? So M must be something to make the marker gene and the antithrombin gene. So it's a promoter region. And then as well as that, N must be a terminator region. So it's important to appreciate that promoter regions tell essentially like yo, RNA polymerase, start making mRNA here, and terminator regions say, all right, you can start making mRNA here. If you don't have these, then RNA polymerase can't transcribe this protein, these 2 actually, because they're both going to be produced as proteins. It can't transcribe it, nor can it translate it either, because if there's no mRNA, no translation can happen, then the gene is not expressed. So this whole topic is hinging on you being able to make the links between transcription and translation in this. So then it says explain the purpose of the marker gene. Well, I just said before, it's like they're putting what have they done? They've put this gene inside goats. So, for example, if it's like in embryos and stuff, we want to know which one's actually done it. So it allows identification of genetically modified like goats or the embryos. So maybe what they did is they put them in like a sperm or something, and then made like fertilized them, and then just looked at the ones that glowed. That's the word, right? That's a weird word. Glowed under UV light, and then we're like, these are the ones. The enhancer only stimulates region M in the milk producing glands of a goat. Suggests two explanations for the importance of the enhancer being produced in the DNA fragment transferred. So, again, what it's telling you is that region M, the actual promoter region, is only active in milk producing glands of a goat, nowhere else. So we need to think, well, what's the protein? It's antithrombin, and it stops blood clotting. So if it's antithrombin and it's only being made in the milk, why might that be a good thing? Well, let's think about whether let's say it was produced throughout their entire body. One, in order to extract this, you would have to collect the goat's blood and then filter and all of that kind of stuff. And like, generally speaking, mammals don't like not having blood. So it's easier to extract from milk versus the blood. And as well as that, if it was in their blood, it's something that stops clotting. So if they've got too much antithrombin, then they won't clot at all and bleed to death. So antithrombin increased antithrombin could prevent clotting, and it could cause bleeding. So remember to relate it back to what the question's saying. The question is just telling you that, like, this thing is only made in the milk glands. Why? And you need to relate it using the examples they've told you. Scientists investigated the effect of full sun and shade on the rate of photosynthesis in a shade-tolerant tree. To estimate the rate of photosynthesis, they measured the uptake of CO2 in a forest. They measured during 2 parts of the day, so from 8:30 to 9:40 a.m., I'm presuming, and then from 11, yeah, because it's in 24, so then 11:40 to 1:15 p.m. And the results have been taken out, soz. So I can't really do any of this, but I could probably sort of give you an indication of what it might have done. So I'm presuming there's a bunch of units to muck about, and it's probably something like micromoles per meter squared. So you probably have to do something like that to get rid of the units to make it micromoles. Figure 8 shows that there's a small difference in the mean uptake of CO2 between 8:30 and 9:40 hours by the trees in full sun and by the trees in the shade. When the scientists performed a statistical test on this data, they got p to be greater than 0.5. State what this p value says. So whenever you're doing this question, please do not say results or the results are due to chance. That doesn't mean anything. What results are due to chance? Because if I flip a coin 10 times and I get 10 heads every single one, and then I say my p-value for me getting 10 times is is higher than 0.5, and then you say, well, the results aren't significant. What is? Me flipping the coin 10 times? Me getting heads 10 times? Like, which bit of it? So if you're going to do this, number one is to figure out what this p-value is actually measuring directly. So again, if you read the question, it says when the scientists did a statistical test on these data, they calculated p equals greater than 0.5. Now, obviously, I can't see the data, but the question tells you enough to work it out. Because figure 8 shows there's a small difference in the mean uptake of CO2 between 8:30 and 9:40 hours. So the p-value, what is it being done on? It must be the difference in mean CO2 uptake between the 8:30 to 9:40 hours. And they've told you that this p-value that's testing this thing is greater than 0.5. Therefore, there is no significant difference in and then this whole thing basically. So the difference in mean CO2 uptake, blah 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 blah. You have to say what it specifically is measuring. But that doesn't tell us what your understanding of the p-value. That's just saying the conclusion. They've told you the p-value is higher than 0.5. Therefore, it's the probability of, the probability, prob for short, of this difference, or the difference that this whole thing basically, being due to chance is greater than 0.5, or in this 0.5 is pretty rough actually, 50%. So it's important. You can't just say the results are due to chance, the results aren't significant, blah 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 blah. You have to say what is not significant or what is significant, and so literally scribble out what the p-value is testing directly. I've seen it come up time and time again. In this species of tree, very high light intensities can inhibit the release of electrons from chlorophyll. Suggest how this explain the results in figure 8 from 1140 to what is it, 13.15 hours. So obviously I can't see the data, but we can still figure out what's going on. So they've said that very high light intensity results in what? Less electron release from chlorophyll. What was the table measuring? The rate, so it's the measured uptake of CO2 by the trees. So what's happened is it's probably, let me think, less CO2 uptake is probably what's happened. If they're gonna, because if you're photosynthesizing like crazy, you're gonna take more CO2 up. So what's probably happened is this. So again, look at the question and think of what the actual stages are of what they've said, like what the gap or the thing is they want you to explain, and then explain it. So suggest how this could explain the results for these. Well, if you have less electron released from the chlorophyll, think about what the whole point of that is. So in non-cyclic photophosphorylation, in photosystem 2, chlorophyll releases an electron. Like imagine if I draw, that's PS2, and there's your chlorophyll, I'm going to write C for short. It releases an electron, it moves down your electron transport chain, and it gets to photosystem 1. And then once it's at photosystem 1, sorry, light shines on that, and then light shines on photosystem 1, and the electron's excited again, and it's passed on to something. What is that? It's NADP. So then you get reduced, I'm going to write red for short, reduced NADP, and you also get some ATP as well through the process, because basically the way I like to think of it, it doesn't really, I don't know if this is technically correct, but it makes sense to me. Chlorophyll, if you shine light on an electron, it gains energy. The energy from the electron, it's been released. It's like, yeah, I'm free to leave. Once it's done that, it can't really get back into the photosystem because it has too much energy, so it needs to lose some. So what happens is as it moves down the electron transport chain, it's losing energy. But energy's got to go from one place to another. It can't just like freely, just disappear. So what happens with that energy is that protons are then moved about like this. And then what happens is they move, so from the stroma to the hydrocarbon and so forth. So then what happens then is that those protons then move through ATP synthase and so forth because they want to, balance out that concentration gradient again, and that's where you get ATP. So, and then also as well as that, you get photolysis. I know we're going a bit beyond the question, but photolysis happens to replace that missing electron to start with, and so everyone's happy. So the reason it's called non-cyclic photophosphorylation is non-cyclic means that this electron doesn't come back to the chlorophyll. It's passed on, it's gone. And photophosphorylation means it's using light to phosphorylate ADP into ATP. So it's in the name. But when you're learning this, just think about the electron's energy and where it's going. That helps make understand a sense of this. Anyway, back to the actual thing. So less energy from high light intensity means you've got less electron release from the chlorophyll. So if you've got less electron in the chlorophyll release, then that means we're going to make less ATP and reduced NADP. That's the whole point of that stage, non-cyclic photophosphorylation. So as a result, what happens? Can't just describe everything. You have to say what the specifics are. So this goes to the Calvin cycle, and what happens? You need to be able to think, okay, what does this directly get used in straight away? Not if you can do that without having to list the whole Calvin cycle out, you pretty much know it. Reduced NADP, the first time these two actually appear, is when you're going from GP to TP. So you are reducing GP into TP. So there's less reduction of the GP into TP, and then you say its consequences. So you need to explain why it leads to less CO2 uptake. If you've got less TP, then that means you've got less, well, what does TP do? It either goes into useful organic compounds or remakes RUBP. So that means you're going to have less RUBP made. That still doesn't explain why CO2 drops. Well, if there's less RUBP made, there's less RUBP to bind with CO2 via Rubisco. So therefore, the CO2 uptake decreases. And that's how you do it. So you need to think of the direct consequences step by step. So literally flowcharting it like that. Less electron from the chlorophyll means that you're going to eventually lead to less ATP and reduced NADP from the light-dependent reaction or non-cyclic photophosphorylation, if I'm being more specific. Then you need to say what the direct consequence of this is. Not just like list everything. Say what the specific thing that these are used in are. So the only point these appear in the Calvin cycle is but to reduce GP into TP. That's still, so we've addressed like kind of this part, but we haven't said why that leads to less CO2 uptake. So you need to think, okay, how does this whole thing link to CO2? Where's CO2? Well, CO2 combines with RUBP, but we haven't got anything. We only got to this stage. So you need to think, well, how does TP link to any of this? Well, TP is, used to regenerate, I should say, regenerate RUBP. If there's less RUBP to regenerate, then there's less to react with CO2, so less CO2 is taken up. And that's how you do that. So tomato plants grow best in high light intensities. To increase the yield of tomato plants, a farmer uses LED light bulbs to provide additional light. The increase in dry mass produced can be calculated this equation, D equals L over 0.4 F, where L is the light used in photosynthesis, and F is GPP to NPP conversion for tomato plants. So table 5 shows some of these values for the LED light bulbs, and then it says use the equation in table 5 to calculate the increase in dry mass produced when using LED light bulbs and give your answer in standard form. So D is dry mass, that's L over 0.4 F, and they've given you the numbers, so they want you to find out. So, okay, if we use the numbers they've given you, so D equals 2.87 times 10 to the minus 2 over 0.4 times 20, we get what? 2.87 times 10 to the minus 2 over 0.4 times 20. Oh, that's all they want you to do. Fair enough. 3.5875 times 10 to the minus 3. So all they want you to do is they just want you to do the calculation here, because this question... Oh, it says, sorry, whoops, D is the increase in dry mass. Misread the question. So all you got to do is just plug the numbers in, and then you've actually got to figure out the units. So already off the bat, it's 3.5875 times 10 to the minus 3. In terms of its units, look at the units they've given you. So they've given you megajoules per meter squared per hour, and we're dividing it by megajoules per kilogram. So when you're dividing like indices, what you're doing is you're subtracting. So for example, if I do 2 squared over 2 to the 4, it's going to be 2 minus 4. So it's going to be 2 to the minus 2. So you can do it that way. Or what you could do is just have a think as well step by step. So this, I mean, first of all, you can see the MJs cancel out. And what we're left with is meters per, sorry, so per meter second square, sorry, meters to minus 2, h to the minus 1 divided by kilograms to the minus 1. So if we're, what that means is per kilogram, you could think of it as like, well, either one, you can just do, you can just do, well, if you think of it, there's no kilograms here, so that's kilogram 0. If you're dividing it by minus 1, you're adding the power, so it's going to be m to the minus 2 h to the minus 1 kilograms. So that's what it looks like, but you've always got to put the first one being like, the one that's not like per. So that's going to be kilograms, m to minus 2, h to minus 1. Alternatively, you could think of it like this. You've got m to the minus 2, h to the minus 1, and that's like saying you're dividing it by a per kilogram like that, because that's what per kg to minus 1 means. If you divide by a fraction, you're flipping it, so you actually end up with m to the minus 2, h to the minus 1 kg, but remember again, that's got to go to the start, so it's going to be kg and all of this. It's like that. So mature leaves from slow-growing, shade-tolerant plants produce poisonous chemicals that are a defense against being eaten by herbivores. Suggest how this benefits slow-growing, shade-tolerant leaves. So, okay, let's think about this. So they make toxic chemicals, and it's to stop them being eaten by herbivores. So slow-growing, shade-tolerant, poison, less herbivore eating. Why is that a good thing then? So, thoughts? You can have a sec to think about it. All right. So, if they're slow growing, it's their shade tolerance. If they're shade tolerant, what that means is that you've got a slower rate of photosynthesis. So, it's to stop them from being eaten by herbivores and stop the leaves being eaten, we should say, really. So they're slow-growing leaves. So if you've got a decreased rate of photosynthesis, then that means that leaves take a long time to grow. So if they're poisonous and they're not going to be eaten, they're going to be eaten less. But that still doesn't explain why that's a benefit. If they're being eaten less, that means they can grow more. But then why do they want massive leaves? It increases surface area for light absorption, because they've told you that they are shade-tolerant plants. So it means that they can survive in shade, but in order for a plant to survive, it needs enough light. So that mean as an adaptation, it must have gigantic leaves. So scientists measure the concentration of poisonous chemicals produced by shade-tolerant plants in a 6 taxa. They compared this with the mean concentration of poisonous chemicals produced by all the plants and the phylogenetic relationships between these 6 taxa, and here are the results. So makes sense of what the results going on here. So concentration of poisonous chemicals produced compared with mean. So we've got a mean of some sort. What's the mean? Sorry. The mean concentration produced by all plants. So compared to the mean, ferns make less, and monocots make less, and magnolids make more, basal angiosperms make less, and more. So it's compared to the mean. As well as that, they're looking at the phylogenetic trees as well. So this is your most recently evolved. That's not. So then it says, a journalist published the following summary of these results. The more recently a shade-tolerant plant has evolved, the greater the concentration of poisonous chemical it produces. So does the data support this? justify your answer. So you got to say your fors and your against. So the fors are what can you see in the data that describes it, and how does that support the statement? The againsts are, well, not really against, but like more limitations. Like, what are the problems with this? Can you actually generalize this statement to everything and things like that? So if we look here in this, they've said that if it's more recently evolved, then you've got greater concentration. So if we look at the most recently evolved one, that's asterids, and these make a concentration higher than the mean. So, that supports it. Asterids a name. Asterids produce more, poison or whatever than mean concentration. So that implies that, it supports it because that's the most recently evolved and they're making more than the average. But if you look at the ones before it, rosids and angiosperms actually make less. So, but what is it? The rosids and angio, I'm going to write angio for short, produce less. So then, as well as that, let's look at the againsts. Obviously, you should try and make a conclusive statement. So usually say your fors and your against, and you can say therefore. My opinion is that it's no, because, although it's that, we can have a look and see the others. So it says, again, the more recently evolved, the greater the concentration it produces. Well, if that's the case, then surely these bars should be going up like this, and it's not. So, we can see that, for example, magmalids make the most. So the highest concentration compared to mean, it was not in asterids, and they're not the most recent. As well as that, the opposite also applies, because if they're saying that, then it means the older the species evolution is, then the less it makes. Well, ferns are the oldest, and they don't make the least. Don't make least. Right? And then what else can we think of? Well, some of them have the same. So, for example, if we look at ferns, these are mostly recently evolved, and these have the same concentration. So they've evolved at different times. And yeah, you can see that in this case, you can't really be like, oh, there's what is it? There's other factors that might be into play. They've only tested it in plants, because the conclusion is the plant thing. So you gotta have to relate what it is to the question. So there's a couple of them. Okay, so here's how I recommend tackling an essay. Number one, if you can hack it, try to be able to do one in about 30 to 35 minutes. I know it says you should spend I don't know, how long is it? 45 minutes? I think if you get really good at practicing, you should be able to do it off the bat, if you really try. Obviously, that's not a set, you must. It's just something that you should try and aim for, because then it means you get faster and more efficient at it. Even if you don't get it, you're still better, than spending hours on it. As well as that, when revising, I recommend you do both of the essays. Obviously, in the real thing, pick the one that's easier, but in your revision, do all of them. Because if you can do if you can think of points to address for every single essay and you've covered every single one, you're probably in safe ground. So the general problem that I see with a lot of this is that people are too vague, or they just spew just information and nothing useful. There's no addressing of the topic. The questions say usually something like the importance of. So the importance means, why is this important? For example, having arms and legs is important. Why? A lot of people will just say, oh, it helps us move, which is true, but you're not telling me the hows. The hows would be, well, okay, you can move, your muscles can contract, and that can let you move and walk about and this and that, blah, blah, blah. And the reason this is important is because we can do things like, for example, go and get food from the store, or drive a car, or go to work. So it's it's putting that sort of, this is why it's important, not just saying this is what happens, the importance is this, but not actually specifying it. Another example would be, I guess, for example, sight. Humans are able to see because why? They have eyes. This is important because what? And people will just say, oh, it helps them see, but that doesn't tell me the importance of it. The importance would be, if we're looking in the context of survival and stuff, it helps them survive and get out of danger. It helps them, look at something and be like, oh, this isn't poisonous, I should eat this. It's safe, things like that. So when doing these, it's generally speaking, what I recommend is you choose 4 or 5 chunky paragraphs, and you say, again, what the thing is, how each of those things directly make it an important thing, and how it makes it work. So when we're looking at ATP in organisms, so generally what I recommend is you should spend about 5 minutes just quickly planning. And by planning, I mean literally just listing out topics you can address. So if we look at the first one, the uses and importance of ATP, we should think, okay, well, what topics do I know ATP to appear in? Literally just think from topics 1 to 8 throughout, one by one, and just scribble down all of them. So if we look at the first topic, that's biological molecules, in which in which ATP itself appears. So that's obviously one of them. Then we need to think where else. Well, it appears in DNA replication. That's that one. Then if we look at cell structure and division. So remember, it's ATP, so it's all to do with just what needs energy. So what things need energy in topic 2? Well, mitosis is definitely one of them. What else can we have? If we look at the rest of topic 2, that's what cell membranes and stuff like that. So active transport needs it. Right, and then if we look at the cells and the immune system, is there anything there? Well, the immune system does need it for like it's for producing proteins and stuff and like B cells and antibodies. So that's a possible point as well, immune systems. Then if we look at topic 3, that's to do with like exchange and transport and digestion. So obviously ATP is used here, so there's digestion and the co-transport of glucose. Then what else have we got? In plants, mass transport in the phloem. So it's literally just scribbling down stuff here as well, because in the phloem you've got those active transport stuff of things like sugars and stuff from the source into the phloem. If we look at topic 4, that's to do with like what we thinking? What is that all on? It's like mitosis, meiosis, DNA, RNA, protein synthesis. That's one of them, and then obviously as well meiosis is another one. And the thing is you don't have to have completed the curriculum to do this. Obviously, you should do some spread across them, but like should be able to practice a lot of these now. ATP in organisms, photosynthesis is a massive one, and also respiration is like another massive one. Wait, not 6. Sorry, 5 is the same thing. Topic 6. What can we think of? Well, nerve impulses. Where's ATP in that? Well, it's the resting potential. And then obviously as well as that, synapses as well need it. And muscle contraction. Then, what else can we think of? Well, there's the second messenger model in adrenaline when it's working on its stuff to make, cyclic AMP and stuff. And as well as that, for example, controlling water potential as well. Potential in the kidneys, because if you look at the ascending loop of Henle, sodium ions are actively transported out. So that's a lot of topics you could talk about. Choose 4 or 5 juicy ones. So it says, in order to fully address the question and reach the highest bands, you must also include 4 topics and their answers. Yeah, so again, 4 or 5 is great. But you want to be juicy with them. You don't want to just say vague things. So if I pick, for example, let me think. So if I pick photosynthesis, whoops. Let's think about where ATP is relevant in photosynthesis. You need to pinpoint where it is. So the ATP in photosynthesis is all to do with the light-dependent reaction, where you've got non-cyclic photophosphorylation. And they need to talk about its importance. What's the point of the ATP? Well, it's for the reduction of GP into TP. And then why is that important? Because it means it can, one, make useful organic compounds like glucose, and also it can regenerate RUBP and it, continue the cycle. So then actually writing the paragraph would be like, one use of ATP is in the light-dependent reaction where it's produced by, and then you would say what happens. So photolysis, the movement of protons, ATP synthase, ADP plus PI, and you get your ATP. And then this alongside reduced NADP go to the Calvin cycle, which is used to reduce GP, glycerate 3-phosphate, into triose phosphate. This triose phosphate is then able to make useful useful compounds such as glucose, which is important for respiration and more ATP generation and the processes which provides energy for things like growth, repair, and so forth. And it also allows for the regeneration of RUBP, which means the cycle can continue making these useful organic compounds. Then say the why is that important, because you still say it's like that's only teasing at why it's important. You need to say like what the consequences of it is. Generally speaking, a good way to get around that is to just say what would happen if you didn't. So if we didn't have ATP to do this, then we wouldn't have useful organic compounds. So we can't convert sunlight into things like, biomass like carbohydrates and stuff, which means food chains would collapse and, organisms would die because there's no primary producers, and then everything would be gone. Obviously, I'd write this in a bit more formally than that. And as well as that, we couldn't regenerate RUBP, so we couldn't continue the cycle, so this would drop as well further. And that means also plants can't respire and they die as well. So it's kind of like, that's the importance bit. This is really important because if we didn't have it, this would happen, kind of thing. Another example could be for the control of water potential. So again, where is the ATP being used? In the ascending loop of Henle in the kidneys, sodium ions are actively transported out of the nephron tubule into the medulla to lower the water potential. Then water moves out at the descending limb and the collecting ducts and the distal convoluted tubule, and is reabsorbed into the blood. The reason that this is important is because if we didn't have this mechanism, then all of the water that that gets filtered out of the glomerulus into the Nephron would be passed into the urine, and our blood water potential would be too low, and as a result, cells would shrivel, and then they would stop functioning properly, and then we'd die because they can't do their thing. So the ATP is important to be able to do, the active transporting of that and everything else. It's again showing that, but you've got to describe the processes. So generally speaking, what I say is, think of it this way: what marks the start of the process? So like, when is the first thing happening of this one piece you've pinpointed in? When is its end? Why? So like, what's its purpose? So, for example, it's to regenerate RUBP and to make useful organic compounds. So that's the purpose part, and then importance as well. So start and sorry, I should say how in the here as well. So the how is the steps. Oh, so GP is reduced into TP with reduced NADP and this and that, blah blah blah, ATP hydrolyzed, all that stuff, and then the purpose and importance like that. That kind of helps. So the next one is the importance of cycles in biology. So if we're looking at cycles, let's think again in each of the topics from 1 to 8. Every single one. So cycles doesn't have to be like the nitrogen cycle and phosphate only. Cycles are anything where you start with something and you come back to it eventually and so forth. So one example already is ATP, because ATP is used, it's hydrolyzed, and then it's recombined again. So it's a reverse of a reaction. So that's one cycle. Next one, the cell cycle, so mitosis. Why is that important? It means you can grow and develop and replace damaged tissues and so forth. The cardiac cycle and control the heart rate. So the sinoatrial node releases an impulse, it spreads across the atria, causing them to contract, and then it goes to the AV node, and there's a bit of a delay before the AV node reacts so that the atria can finish contracting, and the ventricles then contract because it goes down the bundle of His and up the Purkinje fibers. And that means you can have controlled coordinate heartbeats, which means the blood is moved from the atria into the ventricles and out into the circulation. If we didn't have that, blood would just pool and stagnate and you'd die. Other things as well, so meiosis is another cell cycle. It's important to make gametes so you can reproduce and carry on, an organism's lineage. Photosynthesis, the light-independent reaction of the Calvin cycle. Again, it's important to make useful organic compounds. And it's also really important and useful to make RUBP so that you can continue the cycles again and so forth. Respiration, the Krebs cycle. So it starts with oxaloacetate, finishes oxaloacetate. Why is that important? The electron transport chain as well in oxidative phosphorylation. In nutrient cycles, the nitrogen cycle, the phosphate cycles, they're both important to return like material, like nutrients, like for example, nitrates that use in proteins and stuff, back to the soil so that organisms can, continue living and not dying and stuff. Nerve impulses, the action potential is technically a cycle because you've got your big massive depolarization and eventually come back to normal again. Back to your resting membrane potential. Muscle contractions also a cycle because you've got the actin myosin bridges and all that kind of stuff, and then it eventually comes back to the start. Negative feedback is a massively important cycle, so controlling blood glucose concentration because you need enough. You don't want it to be too low so that there's not enough glucose for energy for respiration to provide energy and ATP for cell processes. But you also don't want it to be too high so that the water potential is too low and cells shrivel and die. And as well as that goes for the blood water potential as well. Any of the cycles, for example, predation, predator-prey relationships as well, that's important. Because if you have too many of one or too many of the other, then obviously the population starts to decrease and you get like changes in the population, content and stuff. And then as well as that recombinant DNA technology. So for example, PCR is a cycle process. You just keep doing it again, again, and again, and again, and again. And why is that important? Well, it means that we can replicate DNA fragments. For example, if we wanted to make human insulin genes or something, we could take the fragment, we could replicate it and then stick it in organisms transgenically and so forth and all kinds of stuff like that. So it's again following that structure of what is this one thing relevant to in the cycles, or where, not just the whole process, like where is this thing specifically acting? The ATP, for example, in again the Calvin cycle, GP to TP. Then you need to think what marks the start and finish of it, the how, and then why does this happen? So what's the overall goal? And then the importance. Well, if we didn't have this, this would happen. That generally works. The key thing, though, and my other advice, honestly, is unless you know this topic inside and out and you've read something recently about this that you could say, I'd probably stick to just saying what's relevant to the spec. Because it's better to get 20 out of 25 marks than to try and go for the last 5 and then screw up something and say something terribly wrong and lock yourself out and, down into another couple marks. Because if you do that, you could risk yourself getting locked into like 15 marks, for example, if you say some really, really bad topics. For example, if you look at the mark scheme, it says multi-structural, some significant areas and or more than one irrelevant topic. So if and then if you look at 16 to 20, it says perhaps one significant area and or one irrelevant topic, which detracts from the overall quality of the answer. If you say 2 irrelevant topics and one of them was you trying to shoot for the 25 marks, you've now just locked yourself at 15. So it's just not worth the payoff unless you really know. In my A-level biology, I did AQA, and my question was the 2018 question on the importance, I think it was like interactions in and between organisms. And the only extra thing I wrote off the spec was about blue-footed boobies and their mating rituals. and penguins fighting over each other for potential mates because I saw a meme of it and went and looked it up out of curiosity, like shortly before the exam, like a couple days before. So again, like don't do anything unless you're directly confident it's going to relate. It's just not worth the payoff. Anyways, I hope that helps. Thank you very much. Feel free to drop a comment if there's anything that needs clarifying. I'll try and answer them as best as I can. Please like, share, and subscribe this stuff as well, and help out your friends. I'm going to try and do more of this as well. Thank you.